Hello and welcome to my podcast. My name is Nika and I am an aspiring scientist from the country of Georgia. Today, I am truly honored to host one of the most influential and internationally acclaimed scientists of our time, Professor Zaal Kokaia. He is the head of the Laboratory of Stem Cells and Restorative Neurology at the Lund Stem Cell Center and a professor of experimental medical research at the Department of Clinical Sciences at Lund University. He is the author of more than 155 works and has been published in numerous international scientific journals. His research in the field of medicine examines the process of damaged brain regeneration. Professor Kokaia, thank you very much for joining me today. To begin with, could you share what first sparked your interest in brain regeneration?
Well, in general, to looking back to my story in related to the research, was always fascinating organ. When I started my biology education back in 80s, it was really unclear what I will do in the future, because it was kind of general biology, what was always interesting for me. But with time and after taking the course in the animal and human physiology, today we call it neuroscience, at that time it was animal and human physiology, I realized that it is really something I would like to go deeper and understand better how this system operates. Thinking about brain as a kind of computer which governs the whole our body, all organs and all the physiological processes which is occurring in normal life and during the pathology, was really interesting. And moreover, at that time, my brother, who is 6 years older than me, has already started his doctoral studies at the Institute of Physiology, and I had opportunity to learn a little bit more and to look inside how this work was carried out. And then in the end, I really decided that the brain and brain physiology and pathology, this is something which I would like to work with. I didn't realize at that time that that will be the subject which will take all my time and I will dedicate all my work to this subject. But at that point, it was interesting, and then it became kind of hobby to deal with it, and fascinating. And deeper you dig in the brain, more questions are arising and more fascinated and motivated you become to carry on your research.
That's really fascinating, especially given how complex and mysterious the brain is. Professor Kokaia, what would you say is your main driving force as a biologist?
Curiosity, I guess. Curiosity to understand how our body works, curiosity to understand what is happening in our body and how the brain is governing it. Curiosity to understand how one can repair damaged or altered function, and basically self-understanding and self-learning. That's what is major driving force. And without curiosity, in general, in science, it's very little to do. I mean, you can last for a couple of years, but unless you maintain this curiosity and drive, you can't dedicate whole your life to science. And that's what happens in my case, because what I do, on the daily basis, this is what I like, what I enjoy, and what is motivating me every day to come and do a little bit more and try to understand a little bit more what brain is about.
And Professor Kokaia, what is the main objective of your research? Because you are a very experienced scientist in the field of stem cells. And what's the main objective of your research during these years?
Well, to start with, stem cells was not always my topic. I started my first PhD back in Georgia. I was studying the electrophysiology, intracellular processes of the cortical neurons. We were working on the cats and trying to understand how the inhibitory postsynaptic potentials, the inhibition of the neuronal activity is mediated. What are the ionic and synaptic mechanisms? Then later on, after my PhD, I got an opportunity to visit Sweden in early 90s. And at that time, in the laboratory of Ulla Lindvall, who is professor at Lund University here, is a clinical neurologist, they were studying the epilepsy and epileptogenesis mechanisms. And at that time, there were neurotrophic factors which were recently cloned at that time. And we were lucky to start collaboration with Håkan Persson, professor at Karolinska, who cloned and discovered several neurotrophic factors at that time. And he was a molecular biologist. And we got interested how these neurotrophic factors and their receptors are regulated after seizures in response to the epileptic episodes. And that was what we started to do when I arrived first time in 1990 in October, looking on the animal model of the epilepsy stimulating rats inducing the seizures, and then looking how in these different structures of the brain, neurotrophic factors and their receptors, which were responsible for some plasticity changes at the synaptic and the cellular level, are altered and changed. So that's how we started. And by the end of summer 1991, the paper has been already published in Neuron, looking on this regulation of gene expression in the cortex and in the hippocampus of the brain. From there, we moved to the neurogenesis, because at that time it was clear that epileptic seizures were inducing the production of new neurons in the hippocampus. And there it was really fascinating to see how these new neurons were generated, and they were integrating into the brain, into the hippocampus, and participating into the plasticity and neuronal network. After that, we expanded from the epilepsy and we started to work with the global ischemia, when you include both carotid arteries that induce total deprivation of the blood into the brain, which resembles, for instance, the heart failure when heart stops pumping and then brain gets deprived of the blood flow. And looked on the same thing, how the neurotrophic factors and their receptors are regulated during this insult. And then from the global ischemia, we switched to the focal ischemia, where you not occlude both arteries and globally deprive the brain of the blood flow, but you occlude with embolips, with some filament, the one of the vessels supplying the brain. This was middle cerebral artery. And then we looked again how this insult or brain damage caused by this occlusion regulates expression of different trophic factors and receptors. It was fascinating because at that time, it was not really known whether the stroke has anything to do with the production of new neurons, but our discovery that this lesion in the basal ganglia in the striatum of the rats would cause the production of new neurons, not from the hippocampus, but from subventricular zone, which is another place where the neural stem cells are located in the adult specimens, including rats, and as a matter of fact, in humans. And that was a seminal discovery which we had in, when was it? 2002, we published a paper in Nature Medicine, showing that the stroke will lead to the production of new neurons. The cells which are normally produced from the subventricular zones, they are traveling to the olfactory bulb and making the olfactory bulb into neurons. But what we saw when you induce the damage in the basal ganglia, instead of going to they continue to go to the olfactory bulb, but they went also towards the lesion. And there they differentiated into different type of the neurons, into the striatal projection neurons. And that was really something new. And then there were a series of experiments by others demonstrating that this phenomenon is indeed occurring, not only in rodents, but there are several studies showing also that the same thing is happening in human brain. So from there, this neurogenesis and these adult stem cells led us to the switching to production to another mode of the regeneration, one from the endogenous sources, but another idea we had that these stem cells, instead of forcing the endogenous stem cells to be produced and get them to generate neurons, but also to produce ex vivo in the culture conditions, the progenitors or young neurons, and then transplant them instead. And then that's where we started to use the stem cells. And then from there, my research line is linked to the production and generation of different types of the neurons from the stem cells and transplantation in the stroke, with the aim to restore the neuronal network which is damaged due to the brain insult stroke. So that was the pathway. It was a little bit long answer, but I mean, I just wanted you to take through, so it doesn't happen that you over the sudden start doing this. At the time when I started to work with stem cell transplantation, it was far away from the reality, but nowadays it is a reality also in the clinical setting. So I just gave you the historical perspective how things were developing at that time.
Thank you so much. Professor Kokaia, scientists have been working to advance medicine for years and for decades. And may I ask you, what makes the brain such a difficult organ to repair compared to others?
Well, there are several factors actually in making brain unique from that perspective, from a regenerative perspective. To start with, I mean, brain is not homogeneous tissue. I mean, in the liver, for instance, you have the hepatocytes and then some blood vessels, some other epithelial cells, 3, 4, 5 type of cells. But even neurons, there are dozens of different neural cells in the brain. Moreover, what is unique for the brain, that these all neurons, in order to make function, to execute the function, they need to be connected. And then sometimes this connection is quite far away. I mean, the brain neurons which are sitting in the cortex on the surface of our forebrain and the motor cortex, they send their axons, their wires, communication tool down to the spinal cord, and that connection might be up to 1 meter. So this communication, receiving the signals and sending the signals, makes a brain unique structure. And then number of these neurons, I mean, these are millions and millions, and many of them are interconnecting, and the diversity of these neurons. And by their composition, by the transmitter or the communication molecule which they are using, some of them are excitatory, they are sending the positive signals. Some of them are inhibitory, they are inhibiting, stopping the other neurons to communicate to each other. But it's really very complex. It's like a computer network, which makes it very, very difficult to intervene. And also keep in mind that the brain represents only 2% of our body weight, but it consumes 20% of the blood oxygen and nutrients, which also indicates that this is a very energy-dependent organ, and it is working day and night very hard to maintain the functionality of many different organs in our body. On top of everything, the brain is kind of isolated from the rest of the body with the blood-brain barrier. So the blood cells immune cells which can get into any organ when it gets damaged and get repaired. And brain is immune privileged in a sense that under normal conditions, immune cells cannot get into the brain tissue. And it happens only for a short while when you get the acute damage, or it could be this for a longer time with the chronic disease. But nevertheless, that makes, in addition, this from immunology perspective, brain is also quite different from any other organ. So these are multiple factors which makes it not only very complex organ, but also very difficult organ to regenerate and to rewire and reestablish the connectivity, which is the essential part of the brain function.
And Professor Kokaia, you mentioned about diversity of neurons, and as far as I have read in scientific journals, the most resilient neurons are those with rich and diverse connections. And when a person faces cognitive challenges, our brains uh, do not, our brains operate and they go through a very diverse and linear pathways, and the multiple systems of bodies involved. I think that's also what makes the brain such a difficult organ as well.
Yeah, of course, it is. It is really very diverse and very complex, and especially high integrative system, which is kind of feature of the human. I mean, way of thinking, decision making, high cognitive functions, which we call, including memory and emotions. I mean, all these are really very complex, and there are lots of question marks still, which we don't understand. We learned a lot during the last few decades, how at the molecular level, how the brain operates, how it develops, how it responds to the damage, etc. But there are still a lot of unknowns and lots of question marks. And keep in mind that it's not only neurons which constitute the brain. There are lots of similar amount of the glial cells, which previously were considered as a kind of supportive cell. But now we know that they are really very actively involved in many functions, including oligodendrocytes, astrocytes, pericytes, and then they are really essential part of the metabolism and the functionality. Without them, the neurons will not be functioning, and the glial cells themselves, they contribute and they function independently of the neurons and maintaining the metabolic activity of the brain and many other functions, releasing lots of factors, helping with the regeneration, development, for instance, forming the connections, synapses. Without astrocytes and other cells, it won't be possible, or oligodendrocytes, which wire these connections between the neurons. Without them, the communication between the neurons would not be possible at such a fast speed as it is now. And then brain would, without these oligodendrocytes, to have the same processing capacity, brain should be like 15 times larger. And imagine 15 times larger brain, how competent and how complicated it will become. So all these cells and connections and functionalities, they are interconnected, and all of them make this complex computer which sits on inside our head and governs and makes us humans.
Thank you very much. And when we talk about brain injury, neurology, and stem cell therapy, let me ask you this question: Are there a specific type of brain injury or disease where stem cell therapy has shown the most promise?
Yes, I think the one of the earliest brain injuries or brain diseases, I would say, not injury, it's the Parkinson's disease, which is related to degeneration or the death of very specific type. I told you that there are different types of the neurons, and they are using different molecules to communicate with each other. There is this kind of molecule dopamine, which is produced in one of the part of the brainstem here and down in the brain, not in the main part. And then these are the dopaminergic neurons, which then they send their axons, their communication, to the front part of the brain, to the basal ganglia. And then with the Parkinson's disease, these neurons, which are producing dopamine, they are dying. We don't know exactly why they do, but as a matter of fact, they are gradually, this is called neurodegenerative disease. They degenerate, neurons degenerate over the time, and then this symptoms is occurring. So from regenerative perspective, it's a kind of very convenient model because only one type of the neurons, very specific type of the neurons, are dying in this disease. And the idea in starting from early 70s to early, developed more in the early 80s, actually it was developed by Anders Björklund and Ulla Lindvall, my mentor, Ulla, that if we will take the dopaminergic neurons from the developing brain, like from the fetus, and place it in the animal model where the dopaminergic neurons are dead, if they can differentiate into the mature neurons and they can integrate, by integration I mean establish the proper connections with the rest of the brain, maybe it will compensate for the loss of the original dopaminergic cells. And indeed, I mean, to make a very long and complicated story short, it has been shown in the animal models when you damage the dopaminergic neurons in the rat and then transplant these young dopaminergic neurons into these lesioned animals, they can compensate for the motor function. In Parkinson's model, the coordination and the execution of the movement is altered. And that gave rise to the approach, cell therapy approach, that in humans, in human Parkinsonian patients, similar thing can happen. And indeed, in 80s and 90s, there were a series of the clinical studies here in Lund and in United States also, when they were using in human patients transplantation of the fetal dead, I mean abortion dead human fetuses. They were taking this part of the brain which will give rise to the dopaminergic neurons. They were making suspension and injecting into the human patients, and then that was the proof of principle that it could work. And in many patients, this was really remarkable recovery, even so that they stopped taking drugs, L-Dopa. And unfortunately, this was just proof of principle because you can't use the fetal tissue as a source of the cells, because for one patient there were at least 3-4 fetuses needed, and logistically it was a nightmare to collect all these cells. And moreover, nowadays lots of abortions are done with pharmacologically induced, and then makes it even more complicated to get hold of the fetuses, aborted fetuses. Therefore, stem cells stepped in, and then people started to make dopaminergic neurons, or trying to develop the protocols which will allow from the stem cell to make dopaminergic neuron. Basically, when we talk about stem cells, let me explain. Stem cells by themselves have just two functions. First, that they can divide and reproduce themselves indefinitely. They can divide, and generate new and new stem cells. But if you place them under certain conditions, if you apply certain molecules to them, if they are located during the development into a certain area, they start to differentiate, they start to mature, they start to make cells with certain function. So the challenge was to define the protocol, or to understand how from these stem cells, what we should apply, what stimulus we should give to these cells in order to make the dopaminergic neurons. And indeed, these protocols have been developed over the decades. And nowadays we have the very clearly defined protocols, the ways from making from embryonic stem cells to make the dopaminergic progenitors, young dopaminergic neurons. And as we speak, there is a clinical study ongoing now in our clinic at Lund University Hospital, where patients are transplanted with the dopaminergic cells derived from the embryonic stem cells. So they make this kind of vials, the portion of the dopaminergic neurons from the embryonic stem cells. They test, validate these cells, that they are functional and they can have capacity to integrate into the brain, and then they implant into the patients. It is ongoing study. We don't have the results yet, but soon in a year or so, we will know how. So far, it's going fine, this study. We don't have any complications or any adverse effect, but it is open label study, and then we will know more about the clinical outcome of this. So it becomes reality using the stem cells and stem cell derived neurons for cell replacement therapy in the neurological disorders. And Parkinson is one of the most advanced and most kind of studied model. There are several studies now ongoing also in Japan and in US with the spinal cord injury, where they are transplanting these cells. There were a couple of papers released, published a few weeks ago from colleagues of ours, Hideyuki Okano as the main author on this paper, that I met him in June in Bulgaria at the conference, and he was really excited and working on the final version of the paper during the conference, and it was published a couple of weeks ago. So there is a hope also that for the spinal cord injury, that there is a way, possibilities to regain certain functions, not all of it, but at least improve the condition of the patients with the stem cell transplantation during the spinal cord injury.
Professor Kokaia, as far as I have read, and also as far as I know, a few years ago you had invited two of my favorite and iconic scientists, Professor Gordon and Yamanaka. They were awarded for Nobel Prize in Physiology or Medicine in 2012. So could we talk about their research and work over these years?
Yes, indeed. You might know that when scientists are receiving the Nobel Prize, then the universities in Sweden, since Nobel Prize is coming from Sweden, have the possibility to invite the recipients to give the Nobel lecture at their universities. So in 2012, the Nobel Prize was awarded to Sir John Gordon and Yamanaka, a Japanese scientist who was working in United States and in Japan in parallel. So the interesting part of it, these two scientists, they are two generations of the scientists. Yamanaka published his paper in 2006, and 6 years later he got the Nobel Prize. While John Gordon, the studies which he was awarded with Nobel Prize, he was publishing in late 50s, in early 60s. And then it was very first reprogramming approach, when they were taking the nucleus of one type of the frog, putting into the egg, replacing the nucleus into the egg of the other type of the frogs. And then with this replacement of the nucleus of the cells, they were receiving the kind of clone of the frog from whom they took the nucleus. So that was at that time, very kind of, from today's perspective, very primitive, but it was seminal discovery of the reprogramming, that the information, you can transfer the genetic information from one cell to another cell and reprogram it. In case of Yamanaka, what he did, basically it was really breakthrough in the science, because what he did, by using the virus and virus carrying certain type of genes, he was infecting the cells of the skin, the fibroblasts, and overexpressing or de novo expressing these 4 genes, he managed to reverse the somatic or postmitotic, or the cells which cannot proliferate anymore. He reversed these fibroblasts back to the type of the cells which he was calling induced pluripotent stem cells. And pluripotency means that this cell has capacity to generate any type of the cells which are present in our body. In reality, Yamanaka managed to reverse development from stem cells. Normally it goes to the somatic cells, differentiated cells, but he managed to take differentiated cells, skin cells, fibroblasts, and reverse them back to the stem cells. And he called it induced, because it was forced, in a way, induction of the embryonic stem cells. And it turns out that there are lots of similarities with embryonic stem cells, which you can take from the blastocyst of the embryo, and these induced pluripotent stem cells. Nowadays, we are in my lab also, we are using induced pluripotent stem cells as a source for the stem cells, because it avoids lots of ethical issues of using the blastocyst embryonic stem cells, but also it allows to make the stem cells from different diseases, genetic diseases, and model this disease in vitro in the dish. So you make induced pluripotent stem cells from certain disease, and then from this induced pluripotent stem cell, you generate the type of cells which are affected by this disease, and then you have the kind of diseased cells in the dish, and then you can screen different drugs, you can look for different treatments, understand the mechanisms of the pathology, etc. It's not only for the brain, but any other organ. So what was remarkable, again, coming back to Sir Gordon and Yamanaka, that it was so robust that after 6 years, so many groups have reproduced it. It has proven to be so useful for the science that he was, and that's very seldom that in 6 years. Normally it takes lifetime, like in the case of Gordon, to get the Nobel Prize. But in case of Yamanaka, it was so pronounced, so important, and such a big breakthrough that he was awarded. Sir Gordon, I was director at that time of the Stem Cell Center, and we invited in 2012.
That's what I wanted to say.
To visit our university. He gave the Nobel Prize talk. We had lots of conversations and meetings with him, and it was remarkable. On his desk, he has the framed letter which his science teacher sent to his parents, that they shouldn't. The meaning of the letter was that it's on the internet. One can read it in detail, but the meaning was that they shouldn't waste his time and their time of educating his son, Sir Gordon, that because he has no talent and no possibilities to learn things. And well, sometimes luckily our teachers are wrong, and then that has been proven in the case of Gordon, that he became a Nobel Prize winner. With Yamanaka in 2015, he arranged a very big conference in Osaka on the induced pluripotent stem cells, and I was honored to be part of. I was invited with the lecture to this meeting, and I met Yamanaka only one time there, and it was really exciting meeting. And 3 years after the Nobel Prize, in Japan they created a special institute which he is still heading. And they have been investing in Japan a lot of money and resources in developing the cell therapy based on the induced pluripotent stem cells. And he is still a remarkable scientist, continuing lots of fascinating studies using the induced pluripotent. And then the whole team around him exploring different aspects of potential use of induced pluripotent stem cells as a tool for the therapy and the regeneration, not only brain, but also many other organs in the body.
So, from Professor Gordon's experience, we can say that a pen, a book, and a teacher can change the world.
Definitely. Sometimes you have to go against odds, and then if you are perseverant and you are going towards your dreams, you can achieve many things which might not seem in the beginning that they are achievable.
And Professor Kokaia, as a scientist, what do you see as the biggest challenge scientists face in implementing stem cell therapy for stroke patients? And uh, how is the research going on with this subject?
Stroke is a very complex disease in contrast to Parkinson's disease, as an example, because it's very difficult to find two similar stroke patients because of the location of the damage, which vessel is occluded, because of the extent of the damage, how big the damage is, because of the comorbidities and the condition of the patient, because normally stroke is occurring in the aged patients and they have many other diseases besides the stroke. Therefore, this diversity and the variety doesn't allow really to make it kind of universal for any kind of the stroke patient. So we need to really understand which type of lesion, which type of patients will be suitable and benefit from this kind of treatment. And therefore, this makes it's a little bit more complex to define all these conditions. Moreover, there is also a challenge to make this, to understand when and how these cells should be delivered into the brain. Because you might know that with the ischemic stroke, and that's the dominant, the majority of the stroke patients, they have this ischemia when the blood vessel got stuck. There is another stroke, which is hemorrhagic, when there is a bleeding. So, but we're talking about now on the ischemic stroke. So for the ischemic stroke, there are ways, if the patient is admitted to the hospital quick enough, within 6 hours, if you want to do the certain procedures by surgically with a shunt, removing the clot of the blood and restoring the blood flow, that's very efficient treatment, but only in the acute phase, up to 6 hours.
And also, they use a drug called cerebrolize or something.
Cerebralizing is not really, it's tissue plasma gene activator, TPA, which dissolves this clot. Cerebralizing, this is different story. I mean, this we don't want to go in this part of it. But TPA, tissue plasma gene activator, I mean, this is the substance which can dissolve the. So there are two ways: mechanically removing from big vessels the clot, or in the small vessels. If you deliver this drug, it can dissolve this clot of the blood, which prevents blood from flow and then restores the blood flow. And then, but unfortunately, not all the patients will be suitable for this kind of treatment, and not all the patients will be admitted within this therapeutic window. And the challenge is now in this regeneration phase to understand how and where to apply these cells, what type of cells we need to apply. It takes quite some time. There have been several studies, clinical studies with open label on the safety side. However, we still don't have any phase 2 or even phase 3 or moreover studies where we can say that the efficacy of the cell therapy is working in humans. However, based on the animal studies, we are very optimistic that in years to come, we will manage to develop the proper protocol, which will allow us to help the stroke survivors to improve their quality of life and to regain independence or regain the functionality which they are losing due to the stroke-induced damage. And my group is working on the cortical damage, so cortical area when it's damaged, and trying to produce, reprogram the cells from the skin and make the cortical specific type, not just neurons, but very specific type excitatory projection neurons, which are sitting in the cortex. We started recently also production or generation of the oligodendrocytes because these are also important glial cells which help to myelinate, to produce the myelin or this insulation of the axons, the connecting wires, and for the integration and restoration of the network, they are also very important. So we are working very hard, and we hope that in years to come, we will gradually move towards the clinic. However, before moving to the clinic, now we are also developing a new model because normally you do the experiments in the rodents, in rat or mice. And this is two species. We use human cells and we transplant into the rodent, and then it's called the xenotransplantation from one species to another. And normally there is this immune reaction that the body will reject these cells. Therefore, we are working with the immune deficient animals where the immune rejection is not occurring. But what we are developing recently, so we are taking the cortical tissue from the patients when they are operated for the epilepsy. They are making certain small window in the cortex in order to access the deep structures in the brain. And then this piece of the cortex, which neurosurgeons are removing, we are getting in immediately in our lab and then making kind of tissue culture. So keeping this piece of the cortex in the culture condition in vitro, and then trying to do the transplantation there of our cells and assess their electrophysiological properties, how the cells integrate. We published several papers indicating and clearly showing that these human iPS-derived cortical neurons can survive into human tissue. They can establish synaptic connections and they can integrate into the system quite efficiently. So this is very promising discoveries, which will make it easier to translate, to go to the clinic, to stroke patients and do the transplantation studies there. So we are really hopeful and optimistic that this approach and these strategies will be moving towards the clinic in years to come.
Yes, and I think that we should be thankful for scientists working on this subject. And I imagine overcoming those challenges takes years of careful research and persistence as well. And Professor Kokaia, are we any closer to seeing a stem cell therapy used in real clinical settings for stroke patients?
Can you please repeat the question?
Are we any closer to seeing stem cell therapy used in real clinical settings for stroke patients? So, do you think that scientists will implement a stem cell therapy to treat stroke patients in near future?
Yeah, certainly. As I mentioned before, we are moving there. I mean, slowly but steadily. It takes quite some time to translate. There are lots of ethical issues, lots of regulatory issues, also proof of principle that it will work, or what kind of patients, which patients can be
Ethical
included.
considerations as well.
Definitely. So you don't want, for safety issues, for instance, you don't want these cells, these stem cells, have capacity of proliferating, and then you don't want to cure stroke, but induce the brain tumor or excessive proliferation of these cells in the brain, etc., etc. Toxicology studies. There are lots of steps, which will regulatory issues, which will take some time to move it towards the clinic. But many groups are now moving towards this direction, and I'm really optimistic that in years to come we will manage to do the proper clinical studies and validate the approaches and the strategies which we have been developing in the animal models, that they can be applicable also in the human conditions.
And I'm sure that gives real hope for the future of medicine and science. And in your view, what has been one of the most significant breakthroughs in recent years in science?
In science in general, or what do you mean?
In this field of science, stem cells and neuroscience.
Hmm. Well, I think the fact that we can generate nowadays, we can prime the stem cells towards different phenotype. I mean, years ago we were just happy if we can make the neurons, and later just to make the inhibitory or excitatory neurons. Nowadays we are making very specific type of the neurons with very closely resembling what is in the adult brain. And I think this is the major kind of breakthrough, that you can tailor make the defined type of the neurons, which you can then consider as a tool for the cell therapy in the brain. I think this is the major that we are learning now, and we are capable of producing the very specific type of the not only the neurons but also the glial cells, including astrocytes and the oligodendrocytes from the induced pluripotent stem cells. And I believe that in reality we might face the situation that just replacing the neurons might not be sufficient, and we might need to have more kind of complex approach in order to make this treatment efficient and achieve the goals of the sufficient regeneration of the brain function.
And it's for me, it's really incredible how far the field has come in such a relatively short time. I mean, stem cell therapy and neuroscience in general. And in your scientific career, what has been one of the most unexpected findings in your research?
Um, Coming back to early 2000, I mentioned already this stroke-induced neurogenesis, which we discovered by chance. Actually, we were just looking on the lesion and labeling certain cells, and while looking in the microscope, we noticed that there were some cells which were stained with these young neuronal markers like double coating. And we started to question where these young neurons are coming from in the adult brain. And that led us to this discovery that there is this adult neurogenesis from the subventricular zone in response to stroke. And then most striking was that when we identified these cells, that they are assuming or they become the type of neurons, striatal projection neurons, which were lost due to the strokes, reprogrammed. Instead of becoming the olfactory bulb interneuron, they become the striatal projection neurons. And that was most surprising and most exciting. And this is one of the highest cited my work so far. And that reflects that it was really very important, and there were very interesting finding at that time.
Uh, I'm sure moments like that are incredibly motivating for scientists. Professor Kokaia, let me ask you, what are your long-term goals, both personally and for your laboratory's work?
Well, I mean, my personal and scientific and the lab work, as I told you, to move further with this approach and to dissect the functions which these transplanted neurons can execute. You know, when you transplant the cells, they have dual effect in a way. They have the plastic effect very early after transplantation, because these grafted cells, they can release different trophic factors, different molecules, which can boost the endogenously existing the plasticity and regenerative capacity, which is in the aged animals and aged persons is decreased. The capacity of regeneration is decreased. Of course, younger people have it higher as compared to the aged one. That's the first step. And the second step, it takes a while for these cells to differentiate and to get integrated, to receive the synaptic connections and to send and establish synaptic connections with the host brain. And that's the second stage. Now, the challenge is to understand what kind of the impairment these type of cells can repair at early stage and the late stage. Also, we are working now to understand what are the cognitive impairments with this kind of transplantation one can improve. Because as I told you, we need to really be very kind of personalized medicine, which means that we need to tailor make the treatment for individuals, depending what are the impairments and what are their symptoms, and try to kind of goal direct regeneration by providing brain with the building material, proper building material, and then allowing brain to use these cells for self-repair and regeneration. So this is the major challenge. And also, we are working a lot now trying to understand the role of the immune cells, inflammation, which is occurring when you damage is in place in the brain, and trying to understand this interplay and interaction between this immune system, immune cells in the brain, and transplantation, because we have shown that the immune cells themselves can have very big beneficial effect as well as detrimental. So they have this dual effect. And in combination with the transplantation, how to optimize these regenerative processes, that's another major challenge, which I would like to dedicate the remaining years of my scientific career.
That's very nice of a scientist. And how do you see the future of artificial intelligence in this specific field of science?
That's a very, very good question. At the moment, we are using the machine learning and the AI to train to quantify and to assess the outcome of the transplantation, to count the cells, very, very, very kind of in simple way. So, but what will be used it for in the future, it's really difficult to say in kind of this setting. I mean, there are lots of possibilities in the diagnostics, and probably, as I told you, to identify, to kind of assess the condition of the stroke patients, and to identify, to sort out what type of treatment will be most suitable, or what type of cells, based on this multifactorial input into the artificial intelligence, feeding it with the information about the patient and trying to figure out what will be the. But it's going to take some time, because we need to generate substantial amount of data to feed the artificial intelligence and train it in order to get the feedback from it and then validate the data. Of course, we can't, when it comes to the patients, it's very difficult to blindly trust the computers at this point, artificial intelligence, because they are very resilient in admitting that they make sometimes big mistakes, and they are making up some data. And that's very dangerous part of the interaction with artificial intelligence. You need to be very alerted. And when it comes to the concrete data and concrete information, you need to really validate and check that everything is correct and everything is as provided by that. So I think we are in the transition phase. It's wonderful and excellent assistance for us as scientists, as the clinicians, but at the same time, we need to be really very careful to avoid any mistakes, and it will take some time before we can fully rely and trust this system for when it comes to the healthcare system, because this is you might have some irreversible mistakes. I mean, it's not in animal experiments you can afford some mistakes and some trials and mistakes and efforts, but when it comes to the human being and human life and human healthcare, one should be really very careful to not make some mistakes which cannot be rectified later on.
Yes, and that's how scientists test for vaccines and other drug administrations. Uh,
Definitely.
Professor Kokaia, when we talk about the future of science, we cannot leave this subject without mentioning the importance of young aspiring scientists. And what qualities do you think are essential for success in scientific research? And also, how should young aspiring scientists balance their ambition with patience in this field?
Well, that's a very good point. I mean, people don't go to science to get rich or to have the stable work. It's very competitive nowadays. To establish yourself as an independent scientist, group leader, it takes ages. I mean, after bachelor's, you have to make master's degree, then PhD, then postdoc training, maybe a couple of postdoc trainings. Then you have this kind of transition, young scientist position as a group leader, as a principal investigator, and it takes maybe up to 20 years in order to kind of reach the point when you start doing whatever you are interested in, when you become the group leader and independent scientist. So therefore, you don't go to the research. I'm talking about biomedical research, but basically it is the same in any other field. So therefore, I think you need to really decide that you are really interested and motivated. It takes a dedication to work and persistence. You should never, as I tell my students, you should never give up. If you believe in certain direction, I mean, you might be modern science is hypothesis based, right? So you come up with the hypothesis and then you validate this hypothesis, whether this is right or wrong. On the other hand, people are looking for very exciting data, which they call positive data, but the data shouldn't be positive or negative. The data should be conclusive. Sometimes so-called negative data, which doesn't show any difference between two different treatments, as an example, they might be equally important for the development of the field and to save a lot of money and lots of effort and lots of work. Therefore, what I'm telling my students, so be persistent, dedicated, and aim for the conclusive data. And that's what is important in science. And then you need to maintain your kind of interest and research mind. I don't encourage, I call it jumpers, when some hot topic arises and people start to doing it, and then another hot topic, they jump to another field or another area because it is hot at the moment. So I think one needs to learn from these areas and kind of incorporate, integrate in your main research line, and then persistence and the dedication, it always pays back. And that's what science is about. Keep being motivated, dedicated, and anything is possible. So you will always achieve your goal if you spend enough time and enough efforts in order to achieve it.
That's really great advice, especially for people like me just starting out and aiming for the scientific career in the future. Professor Kokaia, thank you very much for joining me today. Again, it's my great pleasure talking about stem cells and to host you in my podcast. And I'm wishing you all the best and also wishing all the best to our beloved country, Georgia.
Thank you very much, Nika, for having me. It was a pleasure, and I'm really excited having the young people like you from very early age, from scientific perspective, being interested and dedicated to the science. And I wish you all the best, and I'm sure you will achieve your goals and you will become really a good scientist, and it will be really rewarding. Trust me, if you are interested in science and you involve and get involved fully in it, it is really fascinating life and fascinating area of the activity. Good luck with that.
Thank you very much.